Hello, everyone, and a very warm welcome to you. I'm Dr. Lawrence Stephen Fayeun from the Federal University of Technology, Akure. It's a pleasure to be your lecturer for Plant Genetics, CSP 409. So this first session, it's all about building a really strong foundation for what's going to be a fascinating semester. Let's dive right in.
So, what's on the agenda for today? Here's a quick roadmap. First, we'll kick things off with a proper welcome and a course overview. Then, we'll take a little journey through the history of genetics, zoom way down into the plant cell, get a handle on some core vocabulary, and finally, we'll map out your next steps for the semester.
All right, first up, welcome to Plant Genetics. The whole point here is to really set the stage and make sure we're all on the same page, with the same expectations for a really successful semester together. As your guide through this course, my goal is to explore the principles of plant genetics with you. And look, this isn't just some abstract science. It is the absolute foundation of crop improvement. It's how we figure out how to feed a growing planet. This course is definitely designed to be challenging, but I am absolutely confident you're going to find it incredibly rewarding.
Now, it's really important to me that we start on solid ground. That's why this session, and in fact the entire course, is synthesized from our official course outline and 4 core genetics textbooks to ensure a rigorous foundation for the semester. We're drawing from the most established knowledge in our field to give you the best possible start.
So what does a typical week actually look like? Well, we'll have a 2-hour lecture to introduce the big ideas. That's followed by a 1-hour tutorial, and that's where we'll really get practical and get our hands dirty. It's our chance to focus on worked examples and calculations, because genetics is a hands-on science. We'll also break down research papers to see how all this theory applies in the real world, especially when it comes to major tropical and subtropical crops.
Okay, with that foundation set, let's take a step back in time. On to our second section, a history of discovery. You know, to really understand where genetics is today, and more importantly, where it's going, we've got to look back at the key milestones that got us here. You know, our modern understanding didn't just pop up out of nowhere. It started way back in 1694 with early observations from guys like Camerarius, who first realized that plants reproduce sexually. Then, of course, in 1865, you have Gregor Mendel doing his groundbreaking work with pea plants, laying down the fundamental laws of heredity. And here's the crazy part. His work was basically ignored for 35 years. Can you believe that? It wasn't until 1900 that 3 different scientists rediscovered it, and that really launched the science of genetics. The story took another massive leap forward in 1944, when DNA was finally officially identified as the stuff of heredity itself.
Now that we have a bit of that historical context, let's zoom in, and I mean way in, down to the microscopic level of the plant cell. This is the physical place where all this genetic action actually happens.
So, inside a plant cell, you might think all the DNA is stored in just one place, but it's not. It actually has 3 distinct homes. The main one, the big one, is the nucleus. But we also find unique sets of genes in these little organelles called plastids, like chloroplasts, and also in the mitochondria. And trust me, each of these genomes has its own unique story to tell. The nucleus is basically the cell's command center. It's the most prominent organelle you'll see, and it houses the vast majority of the plant's genetic blueprint. This is ground zero for what we call Mendelian inheritance. All those rules that Mendel figured out, this is where they happen.
So how is all of that DNA kept organized? On these structures called chromosomes. Just imagine these incredibly long threads of DNA all wrapped up super tightly around proteins to keep them stable. And the genes are all arranged along them in a very specific linear order. Now, in most of the plant's body cells, what we call somatic cells, these chromosomes come in pairs. That gives us a diploid or 2n number. But in the reproductive cells, the gametes, there's only a single set, which we call haploid or n.
But here's where it gets really interesting. The nucleus isn't the only game in town. Plastids, like the chloroplasts that power photosynthesis, have their own little circular chromosome. And crucially, this genetic material is inherited in a totally different way. It follows a pattern called cytoplasmic inheritance, which almost always means it's passed down from the maternal parent. And this has huge implications for plant breeders. It affects commercially vital traits like herbicide resistance and is key for creating male sterile lines for hybrid crops.
Okay, so we've got the history, we know the location. What's next? Well, to really get a grip on this subject, you've got to speak the language. This next section is all about defining that core vocabulary we'll be using all semester long. Let's start with the big one, the gene. You can really think of this as the fundamental unit of heredity. It's just a specific sequence of DNA at a very specific address. We call that a locus on a chromosome. And its whole job is to provide the instructions for a functional product, usually a protein.
Now, a single gene can actually come in different versions. We call these alternative forms alleles. So, for example, the gene for flower color in peas is always at the same spot, the same locus. But one version of it, one allele, might code for purple flowers, while another allele codes for white flowers. Same gene, just different flavors.
So when we talk about the genotype, what we're talking about is the specific combination of alleles an organism actually has. This is its internal genetic code. So for a simple trait, an individual might have two identical dominant alleles, which we'd write as capital A, or maybe two identical recessive alleles, which would be lowercase a. Okay, so if the genotype is the blueprint, then the phenotype is the finished building. It's the observable characteristic, what the plant actually looks like, its height, its color, its yield. And this is a really crucial point. The phenotype isn't just decided by genes alone. It is always the result of the genotype interacting with the environment.
Let's just make this crystal clear. Over on the left, you see the genotype. That's the internal genetic code, like big B, little b. And on the right, you have the phenotype, the physical trait you can actually see that results from that code, like a black coat color. So let's hammer this home. Genotype, that's the instruction manual. Phenotype, that's what you actually see. Getting that difference is, without a doubt, one of the most important things you'll learn.
And that brings us perfectly to our last section for today, your journey this semester. We've laid all the groundwork, and now it's time to talk about your immediate next steps.
So, after all that, what do you say? Ready to see where you stand? I think it's time we check in with your baseline knowledge. Your very first official activity for this course is a diagnostic quiz on basic genetics. Now listen, don't worry. Seriously, don't stress about this. It is not for a grade. The whole idea is just for us to get a feel for where everyone's at so we know our collective starting point and can build from there together effectively.
And with that, we've wrapped up our orientation. Please remember, this AI-assisted revision was curated by me, Dr. Fayeun, to supplement your reading of the primary texts. Think of it as a tool to help you revise and really nail down your understanding. Make sure you subscribe for the full semester series. I look forward to seeing all of you in our first lecture.